What gives you hope?
We, as microbiologists, maybe as scientists in general, we're creative and innovative people. We've been trained to be problem solvers. All those things can be brought together to help us get through this difficult time.
For people who are in training or considering going into training or considering leaving training, just keep in mind that there are a lot of other things you can do with a Ph.D. or an advanced degree or a bachelor's degree in microbiology.
We're swinging one way. It's going to swing back. It really hurts my heart when I see and hear of people leaving the field. I understand it, but we really need to stop that flow.
There's a lot going on behind the scenes. ASM is careful to talk to anybody who is reasonable of any political stripe, anyone who will listen and who is willing to understand the importance of sciences.
We are very committed to uphold these junior scientists, these young people. And so we are going through with ABRCAMS, and it's going to be a great meeting. I think it says a lot about the ASM to commit financially. No matter what the future holds, we are going to have this meeting.
Welcome to ASM Microbe. My name is Stefano Bertuzzi. I'm the Chief Executive Officer at the American Society for Microbiology, and I am delighted to be here hosting a panel on the future of the microbial sciences and of ASM. And I am joined to do this by two very distinguished panelists. To my left is Dr. Theresa Koehler. She goes by Terry. And she is the Chair Emerita of the Department of Microbiology and Molecular Genetics at the McAdam Medical School at the University of Texas in Houston. And one of the things that I really like to highlight about Terry, apart from her incredible accomplishment being a world expert on anthrax and having studied Bacillus anthracis for so many years, was that she was the first recruit of Dr. Sam Kaplan, a former president of ASM, who started the new department in Houston. And so Terry then became president herself. And so that's also a good way, I think, today of remembering Dr. Sam Kaplan, who passed away a little over than a year ago. So we're delighted to have you, Terry, here. And next to Terry is Dr. Alex McAdam. Dr. McAdam, Alex, is the Director of Infectious Diseases Diagnostic Lab and Associate Professor of Pathology at Harvard Medical School. He is also the outgoing editor-in-chief of the Journal of Clinical Microbiology here at ASM. And in his abundant spare time, he studies in his research program the utility of diagnostic tests for infectious diseases in children and the evolution of bacteria during chronic infections in humans. So Alex, welcome. Thank you for being here.
Thank you.
So you come to us from Harvard Medical School. Anything that happened there recently?
A little bit of action at Harvard. It's been a bit of a time. The administration is after Harvard, and they've canceled a number of grants at the university, including grants held by my friends and colleagues at the medical school and at the Harvard School of Public Health. But I think all of us, even in this very difficult time, are really proud of the way that Harvard is standing up for academic freedom.
Thank you. And so with this context, and thank you, Alex, for all the work that you and your colleagues do at Children's Hospital and in research and in caring for patients. So in this context, Terry, how do you see the picture of science at this moment? You know, there are clearly headwinds, so I want to comment on the headwind that you see, but most importantly, what gives you hope?
What gives me hope? So I hope many of you saw the talk last night, the opening session talk, particularly Dr. Margie Lee. And I'm going to probably in my comments echo some of what she said. So, in times of limited resources, I think that animals, including humans, it causes stress and infighting, right? Competition over resources can lead to really bad things. And what gives me hope is that I like to think that we as microbiologists, maybe as scientists in general, we're creative and innovative people. That's number one. And number two, we've been trained to be problem solvers. And so I think those features are, and plus, the fourth thing I'll say is that our sense of community, and here I'll bring in the ASM, I think all those things, being creative, innovative, problem solvers, people who really feel that they have a devoted and committed community, all those things can be brought together that are going to help us get through this difficult time. So I do have a lot of hope for us as a community, and I know I'm here for ASM, but I will say that ASM really is doing its job well in advocacy and community and collaboration, collaborative science, all those things. So I think that we are very well poised in our situation to get through this together.
Wonderful. Thank you, Terry. And indeed, the talk by Margie last night was absolutely fabulous. I don't know if Margie's in the audience here, but if you see her around, please congratulate her. I thought it was so powerful and inspiring. So, Alex, to Terry's point, let's dig a little deeper, and also thinking about what Margie was saying. As she was speaking last night, I was thinking exactly like Terry, I have to say. However, from your perspective as a clinical microbiologist and thinking how high-tech research has become in diagnostics and in that area, so my question for you is, how do you see, what can we do as ASM and as researchers, as clinicians, to really ensure that we don't miss out, on all the potential that has been developed in clinical diagnostics, which is going in a direction that where you may not be able to do it with little resources. So, what can be done, to have our cake and eat it too?
Well, in clinical diagnostics, I would say there are a couple of factors that are operative that may be less operative on the basic science side. The first is that we've often done our research on the skinny. We do our research using our clinical materials and using the results from our clinical laboratories in good part. So if you're validating an assay that nobody has validated and published before outside of the FDA approval, you might use those data and publish them as a paper. And the other difference, and this is less of a difference because this is true, of course, on the basic research side as well, is that a lot of our research is supported by industry. And that support is somewhat more stable for many people than it has been in the case of the federal funding.
That's excellent. Great to hear, actually. It's inspiring knowing that all this development won't come to a grinding halt. That's what I'm hearing.
Well, yes and no. There is still all that work that's being done by industry, all that work that's being done in the clinical laboratories rests on a foundation of basic science. And so while we're still running now, we still need those basic science advances to fuel the future diagnostics.
Yeah, and to the point that Alex is making, I'd like to call out a study of the national academies, which is quite dated, maybe like 10, 15 years ago, and it's very little known. I don't know why, because I think it's really well done. The title is Here or There. And what it is a study of multinational companies, like most of the diagnostic company, and investigating what's the decision process that they make to decide where to place their operations. And the number one factor that emerges for that study is that proximity to university, proximity to talent, and number two is intellectual property protection, right? I thought it would have been the main factor of that, but even surpassed by that was proximity to talent and to basic science, essentially. So you're right, there could be a lag, and I think that study should be dusted off and reminded at this moment. Terry, you're very focused, and you've been throughout your career and still are on mentorship and training the next generation of scientists. It's a tough time now for people getting into science. So what would you say to the young microbiologists that enter the field now? We just spoke to a few of them earlier on.
I mean, again, I think Susan Weiss last night, and she prefaced this by saying, I don't mean to be trite or whatever, but we all are passionate people. And so that's part of it, right? You have to follow your dream, and times may get tough. I would say that, and this goes back to community, if you're a student, you don't just have your primary mentor, you have your thesis committee, you have other faculty around you, you have colleagues in your field. So this is a time more than ever to talk to people, right? Because we can build each other up. There's lots of advice, there are lots of models out there. If you're a PI or a senior investigator, people are watching you, right? So it's stressful for all of us, but I think that, again, I have hope, we have to have hope, and we have to pass that on to our trainees. I think it's really important. We're swinging one way. It's going to swing back. It really hurts my heart when I see and hear of people leaving the field. I understand it, but we really need to stop that flow, because we all know how important it is to stick with this. We can't give up. And we just have to be careful as role models and as trainees. We have to watch and embrace each other more.
Thank you. Alex, any thought maybe on the clinical side?
Well, I agree with everything that Terry said. And I would just say on top of that, for people who are in training or considering going into training or considering leaving training, just keep in mind that there are a lot of other things you can do with a Ph.D. or an advanced degree or a bachelor's degree in microbiology outside of the basic science arena. So in addition to what Terry said, there's the clinical realm, there's industry, there's communications, there's publications. I'm primarily an administrator at this point, but I'm still an editor-in-chief. I'm still very active within ASM, and I could not do that without the training and the sort of organizational skills that I gained and the scientific skills I gained during my PhD.
Can I add to that for a second? So, yes, everything he said. And also just remember that, again, we're trained to be problem solvers. We're trained to understand how to read the literature, identify a knowledge gap, come up with a hypothesis, a set of experiments to test that, and to fix something or to find new information. And that can be applied to so many areas in life, in vocation. And so, if you're still in training now, you're going to be served well by your training no matter where you end up in your career.
I agree. I think that's very well put.
And, we were just talking with the Future Leader Mentorship fellows upstairs, and I was telling them, bringing them an experience that I had when I was a trainee, and I was interviewing for a job, and I was asked, what skills do you have? And I looked at them like a deer in the headlight, and I said, what skill do I know? I can keep a brain slice in culture for 3 weeks, and I think I'm one of the best people at doing it. Is that a skill? And what I missed is exactly what Terry is saying, that who cares about being able to culture a brain slice? You learn it if you have to. But as scientists, we're training about being problem solver. We're training about thinking, of how to go about something. And to me, and I say it in a constructive way, often we forget to remind our students and our trainee that, we're very focused, because to be a scientist, you have to be very focused, otherwise you can get your stuff done. But sometimes we lose the picture. We drill 3 miles deep, and we just go 3 inches wide, and we don't forget that what we're doing there could be relevant for so many more things, and we get stuck in a rut because of that. So I think the point that you made are really key, and I hope that our programs will help get that across. I'd like to switch gear a little bit, and Terry, you've been president now for a year, and you have a couple of weeks more to go as president. So, would you like to comment on what ASM is doing in going forward, like broader than just training now, but with our strategic roadmap? What is our vision for holding the space for the science in the microbial sciences in this quite challenging environment?
I think now more than ever is a time to be visionary. And what we're trying to do with these 3 new scientific units is to put our amazing minds together to come up with, to identify significant issues in the world and to figure out how microbiological sciences can influence that, can fix that problem or get us to a better place. And so it's really a visionary, and there's many reasons that we're going to have these 3 scientific units, but I think the primary reason is to bring people together with a broad base of expertise and interest to try to move science forward in a way that we direct as opposed to outside people direct, right? So we have a lot of skills and knowledge and insight to bring. And so we're trying to generate that with these scientific units. But at the same time, those units they're not independent of each other. So
Do you want to remind everyone what the 3 units are?
I'm sorry, sure. So the first unit that we've already set up this year, and we're very proud to have done that, is ASM Health, right? So that's the intersection of microbiology and human health. We are in the stage now where we are very close to setting up our second unit, which is applied in environmental microbiology. Okay. For each unit, we have a volunteer chair from our membership, and we have a staff scientific director plus an advisory council of about 10 to 14 people. So with the AEM, we have our chair, Jay Lennon, if he's here. We have him set up. Colleen Kraft is our chair of our ASM Health unit, and we are currently we have a director, Dave Mitten, for our ASM Health unit, and we're looking for our director for AEM, or Applied Environmental Microbiology. So we're very close. We have ASM Health. We're very close with AEM. And the third is a very important and dear to me unit, which is mechanism discovery, or what we used to call basic science, right? So the fundamentals that Alex mentioned are so important to drive translational science forward. And so that's coming on the heels very soon of these other 2 units. And we hope that by the end of this calendar year, we'll have all 3 of these units in place.
Wonderful. And I interrupted you. You were trying to say why these are important. Sorry, thank you.
Yeah. So we really want ASM to be driving science, to not be reactionary all the time, to really be thinking, to be forward thinking in our mission. And so these, again, are kind of mini think tanks, but at the same time, they're going to be driven by the membership. Right? So we have ways, we're envisioning ways that membership can feed into the processes and the thoughts and reports and plans, if you will, of each of these units. And I was about to say that the units are going to have, I hope, be very communicative with one another, because if you go online and you look at the composition of the units in terms of the topics that are listed, which is an endless list, but you can see clearly there's a lot of overlap. Right? And so the idea is that they won't be silos. They will bring like-minded people together, but they will also be very collaborative with one another.
Thank you. And this has been a process in the making, and it takes time to do this thing. So we started actually last year. I see Virginia Miller, our past president, who was here, set up, and the units. And Virginia Miller set up the units. We always get tongue twisted, and we think of the Beta Gal Miller units here, And so I want to also recognize Virginia and thanking her for the great work that she did in getting set up for this. So Terry, you are carrying it, to its completion. And Alex, you arrive, and everything is ready for you as president in 2 weeks. What are you going to do with it?
Well, I think that the units, as Terry said, will be defining the future of microbiology. So we, as Terry said, don't want to be reactionary. We want to be leading. That said, we have to admit what a difficult time this is in microbiology and in science more broadly. But I think that an important thing to keep in mind is that we need to keep going. We need to keep ASM healthy because, again, as Terry said, the pendulum will swing. And when science rises like a phoenix from the ashes, ASM needs to be ready and needs to be there to continue to lead the microbial sciences.
Yeah, thank you, Alex, for saying that. And I would amplify even what you're saying here, because we wouldn't have imagined, actually we did imagine, as I tried to say last night, but we didn't know it would come so fast, perhaps, the need for this. And these units really should be a beacon for the microbiologists that the microbiologists build in saying, we, the microbiologists, think that this needs to happen in our field, right? In terms of where the science is going and so on. And to me, I have not given up. I'm optimistic about this. It's tough right now, but I'm optimistic. And I don't believe that this country, the United States in particular, has given up on the concept of innovation, discovery, exploration. You know, the country of the pioneers of Lewis and Clark have forgotten all of a sudden of all this. It's impossible. It's just in the DNA. It's a country of ingenuity and there. And so I think that there will be a moment of rebuild. And the mistake that we could make, we meaning a scientific community could make, is putting Humpty Dumpty together the way it was. And it was a great system. There's no question. It gave us a lot. But that's an opportunity for updating the system for the future. And so I would hope, Alex, that the scientific units, could be that foundry of ideas and concepts so that when the moment comes, ASM is at the table and saying, this is how we, the microbiologists, envision the system should be working to reap benefits for humanity, to its maximum. So I think that's a very challenging aspect. But the fact that we're setting ourselves up that way, I think is a terrific opportunity. Any reaction, Terry? Anything that you'd?
I would add that if you look at the very nice diagram of the units, which I wish we had here, but we don't. But if you remember it, if you look at it on the website, you can see that to me, it looks like a rotary, right? And so people throughout their research programs or their career may get on and off the rotary and have, their interests may change kind of as a continuum, but also there are ways into the rotary. And so my hope is that this new structure that we have will attract people who may not be card-carrying microbiologists, right? So engineers, neurobiologists, cancer biologists, who all may have programs and interests that touch on the microbial sciences, but I hope that the structure allows them to be part of the conversation.
And to that point, Terry, so spot on. I hope everyone will be able to attend the closing plenary session on Science and Society lecture chaired by Dr. David Relman on mirror life. You mentioned it last night in your remarks, Terry, because that is a prime example of an area of, let's say, of interface microbiology, I would call it, which is synthetic biology, that I think microbiologists should have captured, a long time ago, and now it's exactly the opportunity of welcoming and converging, and the units should really function in that way. That's great. I'd like to ask you in the meantime if you have any thoughts about some programs that ASM has been cultivating for years and that we're going to be carrying over even more this year. And I'm thinking in particular of the ABRCMS meeting that will happen in November in Texas, actually, in San Antonio. So do you have anything to say about that event or?
Yeah, so ASM, like all of you personally, is concerned about funding, right? And for many, many years, we've had this wonderful program. The acronym is ABRCMS, that brings all these students, underserved, many of them, together to explore science and to get career advice and to basically just to lift them up. Although we have some funding challenges or expect some funding challenges in the society, we are very committed to many of our values, one of which is to uphold these junior scientists, these young people. And so we are going through with ABRCMS, and it's going to be a great meeting. So please, let people know that this is happening. It's a terrific place for people who maybe not have come from a background that exposed them to science or higher education beyond college, for sure. And so I just think it's going to be a great meeting, and I think it says a lot about the ASM to commit financially, no matter what the future holds, we are going to have this meeting.
Stefano, if I could just take that in a somewhat different direction. You were asking, what is ASM doing now?
Yep.
What critical functions is it serving that it needs to continue? And I would add to that advocacy. where ASM is extremely active and extremely strong. So ASM has people go up on Hill Day. I think you went to Hill Day, didn't you, Terry? Yep. And I look forward to joining that next year. And then in addition, ASM has commented on the skinny budget that's been proposed, particularly in terms of funding of sciences. And if you don't think that ASM is active in advocacy, it's because you're not getting the ASM Advocacy newsletter. That comes out monthly. It's very good, and it tells you the many, many things that ASM is doing in terms of advocacy. There's a lot going on behind the scenes. ASM is careful to talk to anybody who is reasonable of any political stripe, anyone who will listen and who is willing to understand the importance of sciences. ASM keeps all of those doors open, keeps all of those lines of communication open. So there's a lot of activity there as well.
Yeah, thank you for pointing that. And that was actually my next question to you. So thank you for reading my mind. And if I can, maybe I don't want to pound chest. That's not the intent. But to your point, and speaking to ASM members of the activities in that space, thank you, Alex, for mentioning. But while sometimes in the evening, we look at the day back and we say, so what have you really accomplished today? How have we really moved the needle? We voiced a lot of concerns about this and that, but what have we really done? And that happens. But I want to say that I do take a lot of pride in our advocacy team, and I see JR back there. Talk to him. He's a great expert on how to deal with members in Congress. And, some of the activity that we did, for example, among all the activities, we chose to focus on CDC after the pandemic as a strategic direction for our advocacy because it was so in the eye of the storm, and we know what great work CDC does. And so we're very proud. It's not just ASM, obviously, but we made it a priority. We focused in particularly on the NCZID Center, which is infectious disease and zoonotic diseases. And I have to say that center did not see any budget cuts this year. And that is not, it's a tad short of a miracle, if you wish, but a miracle that happens with advocacy. So, guys, advocacy works. Trust me, advocacy works. And that is an example. And I could bring many others. And ASM is a non-political, non-partisan organization. We really talk to everyone. Last week, Alan Siegel and I, Alan Siegel is our Chief Advocacy Officer, met with the new NIH director, and we had a great conversation with him. We offered ASM to be a resource for gain-of-function research and other things, and it was a very good conversation. And we also voiced our deep concerns and our disagreement with some direction that the NIH is going. But that is important to maintain those relationships and be candid. When there is disagreement, we just voice it, but we need to stay engaged. And so these things are really important. So do follow the advocate, do engage, because it does make a difference. Even if sometimes you feel you're just rearranging gravel, but sometimes you move the boulders, and you don't know when that happens. But if you haven't built those relationships before, certainly the boulders won't move. And so that's the important part.
Can I just add to that
Sure.
a little bit? So if you are active in your branch, or even if you're not active in your branch, I just want to point out that our advocacy team will go to your branch and basically provide a toolkit and information to allow your branch to do advocacy. And I think it's so important. We have a lot of young trainees in our branches, and this is something they can do and learn a lot from and actually make a difference. And so there are resources. If you want more information about whether it's a branch or a chapter or just you can get that information from ASM. They're well prepared to help you through that process.
Larry McDaniel, University of Mississippi Medical Center. So this is great, of course, but last year ASM started a Scholars Program, which specifically focused on, I'm not sure I'm allowed to use this term, DEI. So what is the plans for that going forward?
So all the activities that ASM has been doing in this space are still ongoing, and as Terry was also mentioning, activities like ABRCMS, for example, that were federally funded, which likely will not be any longer federally funded. ASM is footing the bill for those, so the programs are continuing, and we can talk specifically about that program. Yeah.
Well, good morning. My name is Zach Weiss. I'm a PhD student at Texas A&M University, and I'm probably one of the unicorns in the group of graduate researchers who actually doesn't want to go into research or industry after graduation. I'm actually hoping to go into science policy. So as a future science policy developer, somebody working in that space, do you have any words of wisdom as to how I use my skills in the future to make sure that I'm advocating for science and microbiology and wherever I end up?
It was a little hard hearing you, but if I understood the question correctly, is advice for getting into science policy as a scientist? Is that what you're asking?
Utilizing my skills as a scientist.
Right.
Less getting into it than
What
doing
skills? Yeah. Okay. Thank you for that question. And Ray is going to give me 3 hours to respond to this question because it's my passion. I'll try to do it in 40 seconds instead. Look, I think I would put out a table, a 2x4 table or something like this, and think in one column you put the skills that you need to be a bench scientist, and the other one that you need to be an effective policy advocate. And you'll find that some of those are exactly the same, and some others are different. So the one that are exactly the same is what Terry was referring before. So as a scientist, you're trained to be an analytical mind. And if you think scientific problems are complex, like policy problems are also very complex in a different way. And you're trained to go about addressing questions and how to go about that. That's a very invaluable skill. I would argue that other skills that you need in policy that in science are important but are not as essential is, for example, writing. Writing is... I see that Virginia disagrees. I have to admit, Virginia, that writing scientific papers is an art and is very important. And I was kind of decent when I was doing that. But when I moved to policy, the level of writing that you can do is a whole other, it's very different. And I wasn't fully prepared for that. And so, for example, I took a class in writing, especially as I'm not a native English speaker. And so learning how to write has been really important. Speaking, in science and delivering is very important as well. But as a scientist, I was trained, for example, when you have a slide, it's like everything that you have on the slide you want to explain. So in lane 1, I have the control. In lane 2, you see this other thing. You can't do that in policy. And so you need to develop a way of communicating that is different than, because we feel as scientists compelled to be rigorous and explaining exactly everything. And we make everyone suffer with us to the experiment. When you're in front of a senator, you don't want to make the senator suffer through your own experiment, right? And you just need to get to the point. It would be really a longer conversation, but the point here is that you're going to find some skills that are really, really important and similar, and others that are different. As a wonderful, we're out of time, but as a wonderful representative of experience of being on the Hill, would you agree, Terry? Anything that from your advocacy on the Hill that you would like to report back?
I also think the other thing is common ground. We all can find, no matter how adversarial you predict the relationship is going to be or how different you are from who you're speaking with, you can find common ground. And I always try to keep that forefront in my mind. We're all people and we can all relate at some level. So my strategy is find the common ground and then go from there. And be a good listener.
Alex, any thought on that?
I would add that about the writing. I'm very interested in writing and publications. You have used analytical skills to figure out how to write a scientific paper, so you have that metacognition about how to do this thing. Those same skills and how do I do this? What does this kind of writing look like? Will carry over into writing and speaking in the advocacy world as well.
Wonderful. Thank you. Thank you for joining us tonight. And most of all, thank you, Terry. I hope you enjoyed your term as president. And we're not letting you go. So we have a lot of stuff for you. And Alex, thank you for joining us. And good luck with your term as president of ASM. We're delighted to have you. Please join me in thanking them.